One thing that I want to step back and describe for people is, you've mentioned qPCR and PCR generally. So this is the polymerase chain reaction. This is
Yes.
a very commonly used, very powerful technology that's used to do genomics stuff. So it's going to, I think, help people understand other things that I want to talk about. But at a very basic level, for people that don't have a background in the relevant biology here, what is PCR and how does it work?
So PCR is a very sensitive tool. It amplifies DNA by copying it with a polymerase, and it works in an exponent on like a log2 scale. What that means is it doubles every single cycle. So if you have a single molecule in the reaction, after one cycle of PCR, you'll have 2. After 2 cycles, you'll have 4. Then you'll have 8. Then you'll have 16, 32. So this quickly explodes into being able to pick up single molecules usually within 40 cycles of PCR. Now, that being said, whenever you're using things that amplify to that degree, you have to have very, very tight controls because you can amplify the wrong thing. This is the specificity of PCR is very dependent on the primers that you use to pull out what you want. So, for instance, if you're not careful and you design a set of primers inside THC synthase, it's very likely that they're going to amplify CBD synthase, CBC synthase, and many of the other cannabinoid genes. You have to be very careful putting primers in those genes to make sure they specifically amplify the cannabinoid gene that you're looking for. Otherwise, it'll run off and do all types of harm, amplifying the wrong stuff. This is something that I think plagued the whole coronavirus epidemic. And one reason I was really outspoken about it is I saw what was going on. It was like, we could never get away with this in the cannabis field. This is just like horrific what they're doing. And it was that they put PCR assays out there without any controls and without any guidance on what CT they should be using to call a sample positive. So why are the controls really important? Well, PCR gives you a number as to what CT this exponential amplification crossed a threshold of fluorescence. Mm-hmm. When these PCR reactions amplify, you can do some tricks where you make them fluoresce in the process and in concordance to how much DNA to synthesize.
Actually, so Kevin, before we get into this, let's try and give people a very simple cartoon example of this. Yeah. So let's say you and I are both suspects in some crime, and they've got a sample from the crime scene, and they want to see if your DNA versus my DNA is present. How would they do that in a way that makes it unmistakable that it's one of us and not the other one?
So that's a good point. The way they currently do this with the FBI is with the CODIS database system, and they will either use STRs, which is they amplify these short tandem repeats that have variable lengths, and those are multi-allelic loci, and they can use about 13 of those to pin somebody down. But there's a lot more information in an STR. And the reason they went with STRs for some of that is because of the technologies in the past were easier at looking at fragments at a different lengths versus fragments that were the same length with a different base. Technology's changed today where now it's easier to look at single nucleotide changes of fragments at the same length with SNP chips and what have you, but you need more of them. You arguably need about 100 SNPs to really get somebody to have the confidence of 1 in a billion type of confidence to put somebody prosecute somebody. So they might look at 100 variants in your genome. They will use PCR and then probably some either allele-specific PCR method or some other sequencing process to read the differences between those fragments. Because in the case of SNPs, they're usually always the same length assay that you're amplifying. So different than the STRs where you're amplifying and looking for a length change in the DNA where you can run through gel. Here you're looking at point mutations in the genome. So when you want to do that with PCR, which people aren't doing necessarily in coronavirus testing, they're just checking presence, absence of a virus. But if you want to do allele-specific PCR, you've got to be much more careful that you design primers that are very sensitive to amplifying one allele versus the other and labeling them differently accordingly.
Yeah. So what you're saying is, if we're at the crime scene and we want to find if Kevin's DNA or my DNA is present, we can use PCR to do that, to detect the DNA with very high sensitivity. The problem is, as we mentioned earlier, right, our genomes are highly similar to each other.
Yes.
So, most of our genes are going to be exactly the same. So the question is, well, how do you tell us apart? Well, you have to find a gene that's different between me and you, and maybe it's only slightly different. And then you have to use this thing called a primer, which is just like a little piece of DNA that'll stick to that particular region of the genome. And you have to be careful about making sure that primer is specific to the version I have or the version Kevin has, but not both.
Exactly. And this is something that has plagued the virus testing front because they're not doing allele-specific PCR with the viruses, and these are RNA viruses that mutate a lot. So we'll keep hearing about new variants that come out, but they screw up the PCR assays, like the S gene target fails. That's because they didn't design that to be necessarily allele-specific, and not that they should have, but that's just a limitation of them having primers. When SNPs fall under your primers, they don't amplify, right? Unless you anticipate that variant into your primer site. But it just shows you can do allele-specific PCR, and when the alleles change, PCR sometimes fails. That is certainly something we had challenges with when we were designing these sex tests on cannabis. We didn't appreciate how polymorphic the Y chromosome was. So we kept rolling out assays that worked in one genetic population, were readily able to predict males and females. We go into a new population, and suddenly our Y chromosome had a variant under our primers, and our assay was failing and calling males female, right? So we've made 4 different versions of that test now to dance around a variety of variants that are in the Y chromosome that we keep discovering. And now it's no longer a single test. It's like a test that's multiplexed and targets multiple regions of the Y to deal with the fact that the Ys are so different. Mm-hmm. That's a presence-absence test that has to do the opposite of what you're asking. It has to not get tripped up on the fact that we have differences. When you actually want to identify those differences, you have to get a very allele-specific PCR going, or do sequencing of the PCR products when you're done to know to split apart the differences that might exist between me and you.
Mm-hmm. So when PCR testing is used in the context of a COVID test, you get your nose swab. If the virus is inside of you, it'll be contained in that sample. And PCR is going to be good at detecting the presence of that virus because it's going to amplify up some of the genetic material that's there. Can you sort of unpack that for us and help us think about what that means for things like false positive and false negative rates?
Yeah, so it's a really important point in that when you swab your nose, there's a paper out there from Dadu, I think it's D-A-D-O-U-H, that goes through the amount of variance you get swabbing your nose. And it's like 1,000 to 10,000 fold variance swabbing between people. So that's a big problem, because when you want to test for viruses, knowing the amount of virus you have there is kind of a number in space. What you really need is the number of viruses per human cells. So you need the denominator. And some of the first tests that came to market didn't have the denominator. And the way you typically measure the denominator is you amplify a human gene and a viral gene at the same time. And you'll look at the ratios of those CT values.
I see. So what you're saying is when you do the nose swab, it's sort of like sticking a spoon up your nose or something, and you're going to scoop out a different amount of virus for each person. And part of that could just be, you got unlucky or lucky with the scoop and got more. What you want to know is how many viral particles per human cell. So you actually need to get both.
You need 2 numbers. Yeah, you need 2 numbers. And the tests that don't have 2 numbers are really, clinically, that would never be allowed in a CLIA laboratory pre-2020. But I think in the panic of the pandemic, people just ran with the first test that came to market. Now the thing about that market is there's hundreds of different tests in the market. So I don't want to speak broadly about them all. Some of them do have internal controls, and that's what you need. Is you need something that measures that human denominator, so you can gauge how much viral load you have. When you don't have that denominator, you get in all types of arguments with people as to what the CT value should be for cutoff. Well, if you can vary by 1,000, that's like 10 CTs. You're never gonna agree on what the CT value should be.
What is CT?
Oh, that's the cycle threshold at which the signal goes positive in PCR. And so the later it is, the less DNA you have. And the earlier it is, the more you have. And it's on a log 2 scale. So every number represents a doubling. So going from, you go out 3.3 CTs, and that's actually 10-fold differences. And then you go out 6.6 CTs, that's 100-fold. And 10 CTs is basically 1,000-fold.
So would an example here be, let's imagine 2 people that get a nose swab, and there is some viral particles in both of them. One person is legitimately sick. They have lots of viral particles inside of them. And so there's lots of viral DNA there. And in fact, they are sick with COVID. The nose swab will pull out a bunch of viral particles. There will be many particles per human cell, and PCR will be able to detect that quickly. So very few cycles will be able to see it. But the other person could just have, just to use a cartoon example, maybe there's just 1 or 10 viral particles that they sniffed up, not enough to make them sick.
Yes.
But you just happen to scoop those out, and you can detect that as positive if you do many, many cycles of this test, even though it's not enough viral particles to actually be causing sickness. That's
right. So there's a couple of things going on here. There's one, the sampling variance in the nose is so wide that you need an internal control to know it. The second issue with this virus is that it has a very long lifespan in your body after you're no longer spreading it. So the papers will show that you might be infectious for about 7 days, but you could be PCR positive for 70 days. And that's because the way this virus infects cells, it gets kind of, when it attaches to ACE2 receptors, it gets kind of invaginated into the cell, and it stays inside this double wall membrane inside the cell. So that virus, even when it's no longer replicating in your cell needs to die for that thing to go away and your immune system to clear it. And so what we're seeing is this RNA from this virus, based on its unique biology, sits around, hides from the immune system by hiding inside your cells, inside of like almost its own nucleus inside of a cell, what they call these double-walled vesicles. And the immune system can't clear it out, but it may effectively keep it from budding from the cell. Anything that buds out of the cell, it can whack down because antibodies and T cells have figured it out. But it can stay hiding in your cells for months. So we often find people are still PCR positive. This is why the CDC has like a moratorium on PCR 90 days out. Like, don't do it again after 90 days because you'll probably still be positive and you won't be sick. Now, that's not really a analytical false positive by PCR. It's finding the RNA and it's really there, but it's a clinical false positive in that it's no longer a quarantine risk.
Right.
I think that's where the PCR went off the rails, is they conflated analytical false positives with clinical false positives. Because in the past, we would never just use a single test to make clinical decisions. We would say, are you symptomatic? And do you have an antigen test that's positive? And doctors consider a lot of its information in making a clinical call. But in the urgency of the pandemic, everyone just said, screw it, we're going to use PCR as the doctor now. And we're going to call anyone who's RNA positive infectious when they're not, and quarantine, too much of society to the point where we quarantine the nurses who need to be in the hospitals. So it got a little bit out of hand, not handling this kind of long tail of PCR positivity we have with RNA. There are correlates to this that we should learn from in the cannabis industry. When we're testing for pathogens in cannabis, we need to be very careful we don't call dead pathogens a problem, right? So we've designed some tools that try to obliterate any RNA or DNA that's not inside of the cell membrane. Wouldn't necessarily handle this thing with SARS because of the unique biology in SARS, but it does handle a lot of cases where you have dead DNA floating around that PCR picks up that doesn't show up on plates, but we see it. Well, we have tools that erase that DNA because that DNA is very susceptible to nucleases that chew it up, and these nucleases can't get inside of a living cell and chew up that DNA. So we have ways of kind of sorting out this live dead problem in the microbial testing that we do in the cannabis field. But it's an important lesson, is that one, you really got to have these internal controls to know how much pathogen are you measuring in relationship to the cannabis plant. We do that by targeting the cannabis genome with an internal control. And then also, can you sift apart the live dead problem? How many of these pathogens are still living, and how much of it is just free circulating DNA from dead organisms? That stuff you can handle with nucleases.